For as long as we've been telling stories, we've been, well, completely obsessed with one thing. The fountain of youth, it's everywhere.
It really is.
But
I mean, whether it's Ponce de Leon in the Florida jungle or some chalice in an adventure movie.
Or just the latest, $100 face cream.
Exactly. It's the ultimate fantasy, isn't it? The idea that you can stop the clock. Or, even better, turn it back.
Exactly. And look, usually that story sits firmly in the mythology section. But what if I told you that, looking at a 2026 paper by Lana and colleagues, that fantasy is colliding, head on, with biological reality? And the weirdest part, it's not a fountain, it's a river.
That is the perfect setup. And I want to be clear right from the start. This isn't just poetry.
Hmm.
When we talk about rivers in this deep dive, we aren't being metaphorical. We're talking about physical, microscopic streams of genetic material.
Flowing through our bloodstream right now.
Right now. A literal river of youth.
That sounds incredible.
It is. And the whole mission for us today is to understand what that actually means.
Yeah.
We're not talking magic water. We're talking about this incredibly sophisticated biological delivery system.
A delivery system run by our own immune cells.
Specifically, one type of immune cell, the CD4 plus T cell. It acts as this logistics hub for longevity.
So the immune system isn't just for, fighting off a cold.
Oh, far from it. In this context, it's packaging up telomeres, those little protective caps on our DNA, and shipping them out.
Shipping them where?
To the brain, the heart, the liver. Basically to the whole system to rejuvenate it.
Okay, before we get into the nitty-gritty of how, because the mechanics sound mind-blowing, we have to drop the headline number from the source. This is the thing that made me stop and actually read the whole paper.
Right.
When they used this therapy on old mice, what actually happened?
This is the data point that makes you sit up and take this seriously. Look, in longevity research, you're usually happy with a 5%, maybe a 10% bump in lifespan. This therapy, it extended the median lifespan of aged mice by about 17 months.
17 months. Okay, let's put it in context, because for a human, that's not a huge deal. But for a mouse...
For a mouse, that is an eternity. A mouse lives maybe 2.5 years, max. Some of the mice in this study lived to be almost 5 years old.
Wow.
If you translate that to human terms, roughly, you're talking about someone living to be 150, maybe older. And they weren't just alive, they stayed physically young.
That's a staggering number. It completely changes the conversation from, how do we live a little longer to how do we actually change the rules?
It does. And to get how that's even possible, we have to go back to the discovery itself, back to what Lana's team actually saw under the microscope. Because people have been looking at these cells for decades and completely missing this.
Right. So take us into the lab. When you look at a lymph node in a textbook, you see cells, just blobs bumping into each other. What did they see that was different?
Well, normally you focus on the cell bodies, right? You count them, you identify them. But this team looked at the space between the cells.
The empty space.
Exactly. And they saw this extracellular material. Now, usually you just dismiss that as debris, junk from dead cells. But this wasn't junk. It was highly organized. They called it vessel-like networks.
So like little highways between the cells.
That's a great way to put it. They saw these elongated dot-like structures flowing like tiny streams through the tissue. And that's where the name came from. Telomere rivers.
And I'm guessing this wasn't just some weird quirk of a lab mouse.
No, that's the classic mousetrap, right? Curing cancer in mice for the 100th time. But no, they checked human plasma blood from healthy donors and found the exact same structures.
So these rivers are flowing in us.
Right now. But here's the real twist. The thing that makes you realize how fundamental this is. They found them in plants.
Wait, plants? Like my orchid?
Specifically orchids, yes. They looked in the xylem, the tissue that moves water up the stem, and there they were. The same telomeric vesicles.
Hold on. You're telling me an orchid, which is separated from us by hundreds of millions of years of evolution, is running the same youth preservation program?
It appears so. Which suggests this mechanism, this river of youth, is ancient. It's not a mammal thing, it's a life thing. It's like life itself figured out it needs this logistical network to share telomeres and maintain the whole organism.
That is wild. It makes you feel a little more connected to your houseplants. We're all just trying to keep our rivers flowing.
It certainly does.
Okay, so the river exists. It's universal. Mice, humans, orchids. But what is it actually made of? You said telomeres, but it can't just be naked DNA floating around, right? That sounds like a major red flag for the immune system.
You are spot on. Naked DNA in the blood usually signals damage, infection. The body hates it. No, these rivers are made of vesicles.
Vesicles, so like tiny bubbles.
Exactly. Little membrane-bound cargo ships. And inside those ships, the precious cargo is the telomeres.
Let's just pause on telomeres. The classic analogy is they're like the little plastic tips on your shoelaces, the aglets.
Right, they keep your chromosomes from fraying.
And the story is always, you're born with long ones, they get shorter every time a cell divides, and when they get too short, the cell gets old and you get old. It's a one-way street.
That's the canonical view, yes. Telomeres as a strict internal clock, a countdown you can't stop. But this research just completely flips that idea on its head.
How so?
In this world, telomeres aren't just a clock you're stuck with. They're a transferable good. They're currency.
So a cell can just Venmo me some telomeres if I'm running low.
Essentially, yes. That's the breakthrough. Cells can donate them to other cells.
Okay, so who are the players in this economy? Who are the donors and who are the distributors?
Let's cast the characters. You have two main immune cells. First, the antigen-presenting cells, or APCs.
Yeah.
These are the scouts. They find threats. Think of them as the donors.
They're rich uncles.
The wealthy benefactors, yes. Then you have the CD4 plus T cells. They are the recipients who then become the distributors. And it is crucial to say the paper is very clear this is a CD4 superpower. CD8 plus T cells, the killer T cells, they can't do this.
It's a specialized job, so how does the transfer happen?
It happens during what we call the immune synapse, the handshake. So an APC meets a T cell to show it a picture of a virus it found.
Look out for this guy.
Exactly. But during that handshake, the APC does something amazing. It packages up telomeres into vesicles and literally pushes them over to the T cell. It's like saying, here's the target, and by the way, here's a bunch of extra life force for the mission.
So it's a refueling station.
It is. The T cell takes them in, and its own telomeres get longer. It becomes a long-lived memory cell. But here's the brilliant part. The APC gives the T cell too many telomeres.
A surplus.
A massive surplus. Way more than the T cell needs, and this is where the river is born. When that T cell divides to build an army, it undergoes something called asymmetric division.
Asymmetric division. Sounds like the Sorting Hat from Harry Potter.
In a way, it is. It divides into two different daughter cells. One becomes that long-term memory cell we talked about. It keeps just enough telomeres for itself. But the other daughter cell, it takes all that massive excess of telomeric DNA that the APC donated, and it packages it back up into new vesicles. And then it releases them into the bloodstream.
It's regifting. It's taking what it needs and sending the rest out to the rest of the body.
That is the perfect analogy.
Yeah.
It's a recycling program. The T cell becomes a factory for these vesicles, and that's the river. It flows out of the lymph nodes and into circulation to help other tissues.
That's incredible. The immune system as a supply chain for repair kits, but there's a catch, isn't there? I remember reading that there's a strict quality control process.
A very strict gatekeeper. And this is where the metabolism science gets really fascinating. Because for this to work, the T cell has to be using the right kind of fuel.
We're talking about burning sugar versus burning fat.
Exactly. For a T cell to create a river that actually rejuvenates things, it has to be using fatty acid oxidation, or FAO. It has to be burning fat.
So burning fat isn't just, good for my summer body. It's essential for my immune system to make these youth particles.
In this context, absolutely. Think of fat burning as the clean energy for the recycling plant. When the T cell burns fat, using a protein called CPT1A, it produces these perfect, clean, effective vesicles.
And what happens if it's burning sugar instead, which is glycolysis?
That's the problem with aging. As we get older, our T cells get tired, they become senescent, and they switch their metabolism, they start burning sugar, and when they do that, they produce a specific enzyme, GAPDH.
GAPDH. Okay, flag that name. This is the villain of our story.
This is the primary antagonist. If GAPDH gets packaged into those vesicles with the telomeres, the magic is broken. The source is very clear. For a river particle to work, it must be GAPDH negative.
So if I'm an old mouse, my T cells might still be releasing vesicles, but they're polluted.
Polluted is a great word for it. They're polluted with this enzyme. And the presence of GAPDH seems to completely negate the rejuvenating effect. It turns the message of youth into just static.
Which explains why just having T cells isn't enough. They have to be metabolically healthy T cells.
Correct. The quality of the fuel determines the quality of the product.
So we need these clean GAPDH-free rivers. What else is in the bubbles besides the telomeres? Because telomeres are just the caps. They don't have instructions, do they?
Right. The telomeres are the raw materials, but the vesicles also contain the instruction manual. They're enriched with what the paper calls stemness factors.
Stemness factors. That sounds like something you'd see on a very expensive skin cream.
It does sound like a beauty product, now with 50% more stemness. But biologically, these are incredibly potent signaling proteins. specifically things like Notch, Wnt, and Runx2.
Okay, I recognize Notch and Want from developmental biology. Those are the signals that tell an embryo how to build itself.
Exactly. They're primal signals that tell a cell, hey, grow, repair, act young. So the T cell is sending out a package with both the raw materials, the telomeres, and the instruction manual, the stemness factors, to tell the cell what to do.
And it lacks the junk like GAPDH. It's a perfect rejuvenation kit.
It is.
So Lana's team figures this all out. They map the river, but then they have to prove it does something.
This is the big experiment, right?
This is it. They took these purified river vesicles, the clean GAPDH negative ones from young, healthy T cells, and injected them into 20-month-old mice.
Which is grandpa age for a mouse.
It is. And they didn't just see one organ get better. The results were systemic. They saw reversal of aging markers in the brain, the liver, the lungs, the kidneys, the heart.
The brain and the heart. Those are tissues that are notoriously hard to repair.
Exactly. And they measured Snellen's markers, proteins like P16 and IL-6. These are the flags that cells raise when they're old and tired. Those markers just plummeted. But more than that, the telomeres inside the cells of those distant tissues actually got longer.
So just to be clear, the T cell mailed the telomeres to the brain, and the brain cells actually got the package and installed the new parts on their own chromosomes.
That's what appears to have happened. The T cells acted as a remote maintenance crew. They delivered the spare parts, and you could see the effect on the mice themselves. What did they look like?
Better fur, more lean body mass, they were more active. I mean, they looked younger.
And as we said at the start, they lived a lot longer.
17 months longer on average. And they contrasted this directly with mice that got old serum or vesicles that were polluted with GAPDH.
And those didn't work.
Zero effect. It proves that the purity of the river, the absence of that glycolytic enzyme, is absolutely everything.
This brings up a really interesting question they asked in the paper, which is, do we even need the T cells? If we know the formula telomeres plus stemness factors minus GAPDH, can't we just brew this up ourselves?
This is the artificial rivers part of the study, and honestly, it's one of the most exciting parts for the future. They decided to bypass the middleman.
The T cell being the middleman.
Right. They went back to the source, the APCs, the donors.
Right.
And they used a genetic trick, Serne, to just silence the GAPDH gene. They basically forbade the cells from making the villain enzyme.
They shut down the pollutant at the factory?
Precisely. And the vesicles these APCs produced, the artificial rivers, they look just like the natural ones. GAPDH negative, loaded with stemness factors.
And did they work?
They worked even better in some ways.
No way, better than nature.
In terms of telomere elongation and the physical appearance of the mice, the results were incredibly strong. And just think about what that means. You might not need a young donor. You wouldn't need to harvest T cells from a teenager to help an older person.
Which avoids a whole weird sci-fi dystopia situation.
Exactly. You could potentially manufacture these youth particles in a lab, in a bioreactor.
That completely changes the scale of something like this. It goes from a custom procedure to an off-the-shelf drug.
Which brings us to the so what of all this. Beyond the mice, beyond the orchids. This research just fundamentally shifts how we should view our immune system.
Yeah, I mean, I always think of T cells as soldiers, the infantry, fighting off the flu, killing cancer cells. They're the Department of Defense.
That's the 20th century view. This paper suggests they're also the Department of Infrastructure, the logistics crew. Lana calls it a quorum sensing system.
Quorum sensing. That's a term from bacteria, isn't it? Waiting until there are enough of you to act.
Yes. But here it implies the immune system is constantly checking the health of the entire community of cells in your body. It's coordinating this signal that says, we're young, keep repairing, keep going.
So aging might not just be random wear and tear, it could be a breakdown in that communication.
Exactly. The signal gets jammed, the T cells get old, they start burning sugar, the GAPDH pollution gets into the river, and the message just stops.
It's a supply chain crisis. The trucks stop running.
That's a profound way to put it. And it's so different from other anti-aging ideas like parabiosis, sewing mice together.
It's vampire science.
Right. That's messy. It's about diluting bad stuff. This is additive. You're adding a specific therapeutic particle. It's precise.
And it's not like CRT cells that go and kill zombie cells.
No, that's a seek and destroy mission. This is seek and rejuvenate. These rivers don't kill old cells. They give them the tools to become young again.
Which is a much friendlier way to do medicine. Here, have some telomeres, perk up.
It's a shift from reactive to proactive maintenance.
So to wrap this all up, we've gone from a handshake between two cells to a literal river of DNA flowing through our bodies, carrying the signal of youth to every organ.
A signal that is totally dependent on metabolic health, on burning fat, and keeping that villain enzyme, GAPDH, out of the system.
And we know it works in mice, extending their lives by a huge margin. We know it exists in humans. We even know it exists in orchids, which is just mind-bending.
It really is.
So here's my final thought, something for you listening to mull over. If our immune system is responsible for keeping us young through these rivers, and this mechanism is conserved all the way down to plants, what does that actually say about aging? We think of it as inevitable decay, a car rusting.
Entropy. It's supposed to be
Okay.
inevitable.
But maybe it's not. Maybe aging is more like a supply chain disruption, a logistical failure, a communication breakdown in our immune system's logistics department. And if that's true, and we can now create artificial rivers, how far are we from a youth transplant that requires no donor at all? Just a vial of engineered vesicles that tells your body, hey, the supply chain is back online, we're back in business.
It poses the ultimate question. Is the limit to our lifespan a biological rule, or is it just a logistical problem we haven't solved yet? And it really seems like we're starting to figure out the logistics.
A fascinating possibility. Thanks for diving deep with us today. Stay curious.